Hi, ladies and gentlemen. I'm going to walk you through how to do the Pastogenics lab activity. I just want to point out what our learning objectives are for today. Hopefully you guys have read through. Okay, it says that how chromosomes and their associated genes are passed from one generation to the next. So that's one key thing we're going to be looking at today. Second thing is that we get 2 copies of each chromosome in our cells, or we have 2 copies in our cells. Those are our homologous pairs. And that as a child, hey, when a child is conceived, they get 1 copy of each chromosome from each parent. But what copy they get is random, right? So it's random whether they get, the maternal copy or the paternal copy of that chromosome. Okay, a child won't look exactly like that individual's brothers and sisters because that child has a unique combination of mom and dad's genes. And each baby will get a unique combo. So we are kind of using this activity to bridge us between looking at meiosis and really looking at inheritance and how traits get passed down from one generation to the next. Hey, if you need a review of meiosis, you can take a look at slide 3. Okay, so feel free to pause right now if you want to read through that, okay, and really take a look at all the different labels on here. Just remember, we start in meiosis with 46 chromosomes, okay, we end with 4 cells that have 23. One of these cells will become a cell that will fuse with, if this is a sperm cell, will fuse with an egg, okay, and fertilization to form a zygote to form a baby. Okay. And then if you move on to slide 4, first of all, if you want more information, there is more information linked here and here in the yellow boxes about the law of independent assortment and the law of segregation. You might need some extra info when we get to our conclusion, okay, but I'm going to briefly explain. Okay, the law of independent assortment says that our homologous chromosomes in metaphase 1 of meiosis, those pairs can line up however they want, right? So our first chromosome pair has the blue on the left and the red on the right. Okay, the second pair, we look at it, it's set up the same. Okay, the second pair has blue on the left, red on the right. Okay, but on the right-hand side, okay, that second pair is flip-flopped, right? And so it's completely random which way each pair lines up. Okay, and the end result, if we look down at the bottom, based on how they line up, we get different combinations of genes in our gametes or in our sex cells, depending on how they line up in metaphase 1. Okay, the law of segregation has to do with individual chromosomes. If you guys remember last week, we talked about in metaphase 2, individual chromosomes will line up down the middle of the cell. And then in anaphase 2, those individual chromosomes get pulled apart. Okay? And when they get pulled apart, they're getting pulled apart into their sister chromatids. Okay? So one sister chromatid is going to the cell that's on the left, the other sister chromatid is going to the cell that's on the right. So they are segregating the sister chromatids or pulling apart those sister chromatids. And that's what the law of segregation says, is that those sister chromatids won't stay together. They're actually going to be split up. And that happens in meiosis too. All right, now moving on to our procedure for today. There are 6 steps today. I think it's, what I would suggest you guys do right now is pause the video, read through these steps. Okay. Then what I'm going to do is actually show you how to do each step. Okay. So as I do each step, you should also do that step. Hey, please do it for yourself. Part of it is going to involve flipping this little coin. I have this simulation off to the side here. Hey, so part of it is going to involve flipping that coin. So make sure you have that app up. I set it up so it was right next to my slide, so it was easy to kind of have them side by side. Okay, so from here, hey, read through the procedure, hey, and then let's look at what we're going to do together on slide 6. Okay, so hopefully you've had a chance to read through all those procedures. Okay, I think it sounds maybe a little more complicated than it is. If we look here, we have grandmother and grandfather 1, grandmother and grandfather 2. Okay, grandmother and grandfather 1 have a daughter, okay, that's going to become the mother of this child. Okay, grandfather to grandmother 2. Hey, they have a son. Hey, and that son becomes the father of this child. Okay, so we are essentially tracing the inheritance pattern of these genes, and our genes are pasta shapes this time through in our activity. Okay, so we're looking at how are these genes passed from the grandfather and grandmother down to the child. Okay, so kind of following from one generation to the next generation to the next generation. Now, you guys will notice in the grandfather, there are outlines that are purple and outlines that are green. And you'll see that in all of the grandparents. Those outlines actually talk about like the great grandparents. So this grandfather, hey, the purple ones and the green ones, hey, they tell you, hey, whether purple is going to be maternal, okay, and green is paternal, right? So that means these purple outlined pasta noodles came from the grandfather's mother, and the green ones came from the grandfather's father, so the great-grandfather. Okay, so let's get on to how to do the activity. Okay, so each one of these shells, right, there's a purple and a green penne pasta shell. I'm going to come over and I'm going to hit flip it. Okay, when I hit the flip it, you'll see I get either a heads or a tails. Hey, this is tails. I come down to my code down here at the bottom of my page, which, by the way, Flip a Coin is the app you're going to use to do this flipping. It's on the bottom left hand in blue of this page. Okay, so tails means that the paternal copy was passed down. Okay, so the paternal copy has the green outline, so I come up to the penne pasta and I drag down the paternal chromosome, okay, for the penne pasta. I'm going to do the same thing for all of the other types of pasta. So again, I'll hit flip it. Okay, I got tails again. Hey, I was looking at the shells this time, so I'm going to move again the maternal copy of that shell gene down to the mother. Okay, so that got passed on. Then I'm going to look at the bow tie, hit flip it again. Okay, and okay, you got tails. You're welcome to do this with a real coin. It might be faster. I don't know. Hey, but if you don't have a coin, the app works great. Okay, so tails again. So another green copy. Hey, another. I said tails is paternal. I'm sorry, I was saying maternal, I think. Okay, and then the last one is the elbow noodle. Okay, so I flip again. And for the elbow noodle, it came up heads. That's the maternal or the purple outline. Okay, so now all of the genes, okay, that I've collected here in Mom's cup, okay, these all came from grandfather 1. So these were associated with the sperm, okay, that then fertilized the egg that came from grandmother. Okay, so we are going to get the genes from grandmother. Same way. Hey, again, I'm going to use the coin flip. Okay, I'm going to do that, and I'm going to pause the video here so you don't have to watch all that flipping. Okay. All right, so as you can see, the mother inherited brown chromosomes from the grandfather. Okay, she inherited blue chromosomes from the grandmother. They were randomly selected with this heads or tails app. Okay, and then I do the exact same process of randomly selecting chromosomes for the father, from grandfather 2, okay, and from grandmother 2. Okay, and you'll notice the chromosomes from grandmother 2 were the 4 black chromosomes, from grandfather 2 were the 4 yellow. Okay, so these are the parents, right? They get married, they have 4 kids. Okay, we are going to create one child here. Okay, so I'll show you guys kind of what it looks like for one kid, and then you guys can go through and figure out the process. All right, ladies and gentlemen, we're back. Okay, so now if you take a look, okay, I have transferred chromosomes from Mom. Okay, Mom is going to pass down one of her 2 copies of each chromosome to her child. Same thing with dad. Hey, dad had 2 copies of every chromosome. Hey, one of those 2 copies got passed down to the child. Hey, and now this child has 2 copies of the elbow noodle gene, 2 copies of the shell gene, 2 copies of the bow tie, 2 copies of the penne pasta gene. Hey, so from here, hey, you guys should take a picture of this. Hey, so I'm going to take a little screenshot of this section. Hey, once you get a screenshot of that, hey, I've got my picture. I'm going to move to slide 7. Hey, and in slide 7, then I'm child's genetic makeup. Hey, I'm going to move that cup so that it fits into this box, and I can resize it a little bit so that it fits in there and doesn't disrupt the others. Then I'm going to answer these questions. How many chromosomes originated from grandfather 1? Okay, well, if we remember back, grandfather 1 was brown, so there's 1 from grandfather 1. Grandmother 1. From grandmother 1 were the blue chromosomes. There were 3. Okay, grandfather 2, okay, was yellow. There were 4. And grandmother 2 was black, and there are 0 of those. Hey, how many total genes then did this child inherit? Well, this child got 2 copies of each, got a total of 8 chromosomes, okay, or 8 different pasta noodles. And that means that there are 4 pairs, right? And 1 copy of each of these is from each parent. Okay, when you go back and look then, and we compare kind of the child's genetic makeup to like the grandparents, okay, the child may not, necessarily get information from a grandmother or a grandfather. More than likely though, because we have 23 pairs, we don't just have 4. Okay, we would probably get at least 1 of our chromosomes from like that grandfather or grandmother generation. Okay, but this is an example where the child didn't. Okay, you're going to go through and follow this same process for 3 other children. Make sure you name your kids. Okay, so mine is Zeke. Probably not spelled like that though. Okay, so that's my pasta child, number 1. Okay, I'm going to go back and do that 3 more times. Well, to do that 3 more times, I have the same parents, okay, but each child is going to have a unique genetic makeup. Okay, so I'm going to transfer these chromosomes back. All right, ladies and gentlemen, I hope that helps you guys figure out how to do the lab.